So let's see, we're continuing on prokaryotes, and we had just finished with magnetosomes, right, that may actually be an organelle in prokaryotes, which by definition prokaryotes usually don't have organelles, right? Only eukaryotes do. So did anyone actually read that section? It's kind of cool, huh? Like having, like I said, like having their own little compass inside of them. So how these line up too is determined by what in the cell. How is it these vesicles that contain iron, how do they line up in that cell? What lines them up in the cell? And you don't have to be specific, you can be general. How do you think structurally we can get stuff to line up within a cell? What do you think, Lance? Poles? Right. Well, think back to eukaryotes for a second. How do we get chromosomes to move from one side to the other? The spindles. What are those spindles made out of? At the molecular level, we have 4 choices for macromolecules, right? Proteins. Remember I said about them being like the workhorses? Like most of the time, like if you say protein is probably the answer. Right? Yeah. They're very important structurally and functionally, and we're going to start looking at and talking about some of those today within the cytoplasm. Are you? Okay. All right. So the cytoskeleton is a series of internal proteins that assist in keeping everything in or moving it to the right locations within the cell. So how do we line those up? It's the proteins within the cell. And we already know this. We know a lot about the proteins in eukaryotes, right? Because eukaryotes are much larger. They're more complex. They have, you know, the spindles during mitotic division and microtubules and actin filaments, right? And especially if you've taken anatomy and physiology, you're like, oh, yeah, actin, oh, God, right? You're having actin nightmare flashbacks to muscle movement, huh? So for the longest time, again, when I was sitting in your guys' seats, we didn't talk about this in microbiology because we really just didn't have the tools at the time to know that these proteins were there. But if you think about it, it's a cell. It's got to organize stuff. It's got to move stuff around. So it's got to have some type of cytoskeleton. And in fact, they do, right? We have lots of evidence now of a cytoskeleton within prokaryotes, and these are some bacterial examples. And so, of course, they've got these funky abbreviated names, right, that kind of freak us out. But by no means do I want you guys memorizing these. I just want you to know in general that they're there and what they do. So, of course, they help with cell wall synthesis. And when those cells are getting ready to divide. So they have this special ring that forms, this Z ring, that literally pinches the two of them apart from each other. And again, similar things happen in eukaryotes. We can see this, and we could see it much early on with just a light microscope, right? In biology class, you guys get to look at mitosis of plants and animals using a standard light microscope. You know, it was more high-powered using things like fluorescence to be able to label these proteins, and that's what they did here. This is still a light microscope, but again, a much more advanced one where they're using fluorescence labels to be able to see these different proteins, right? So they're able to color them this pretty greenish color. So remember, all our electron micrographs, all our pictures made by a microscope, they tell you what type of microscope in general. So FM, remember, is what? Fluorescence microscopy, right? So one of the proteins, the MREB, can associate in a way that it forms these coils within the cell. And so the rod-shaped bacteria, right, and what would be the term for rod-shaped, but we don't tend to use it, bacillus. So in order to actually have this shape, you have to have this protein in this coiled manner inside the cell. Otherwise, it just becomes round. Like, round is like default, like the spherical shape is default. Right? But when you have this protein, because they know this because they've engineered ones without this protein, and guess what? They're round. They're spherical. Right? But the ones that have this one are able to form that rod shape. And so we know that definitely this protein is contributing to the shape in addition to the cell wall, right? Which helps hold that rigid structure of the rod. So we know definitely eukaryotes. We're going to talk about eukaryotes next week. Archaea, remember, also fall into the category of prokaryotes. And again, their cytoskeleton has homologs, similarities, to both bacteria and archaea. And then even still, right, this particular protein, TA0583, oh Lord Jesus, right? Couldn't we just come up for a name with that one? With all these letters and numbers. They probably stand for something, right? They're very similar to actin in eukaryotic cells, how actin works. Others have proteins that closely resemble proteins found, of course, in bacteria. So archaea and bacteria are very similar, and then both of them, you know, seem to have these kind of precursors, you could say, to the protein structures that make up the cytoskeleton that's pretty well defined in eukaryotes. So moving stuff around. So although we don't have several sets per se of chromosomes, usually in a bacterial cell, they will tend to have additional pieces of DNA known as plasmids, these circular pieces of DNA. They're not required, but some, and actually in fact a lot of bacteria have them. But when the cell divides, how do they divide up these structures, right? How do they get one plasmid in one half of the cell and one in the other? Well, these proteins. You know, again, not all that different from what we see what happens in mitosis and meiosis in eukaryotes. These proteins, in this case, actually push them apart from each other to either side of the dividing cell. And again, that movement, that lining up of those magnetosomes in this organism, in this prokaryote, is due to those proteins lining them up, organizing them in that nice pretty line that make that a functional organelle. Gives that cell its polarity, right? Being able to point north like a compass does. Also in the cytoplasm for archaea, remember we said that the nucleoid is the region where the DNA is housed within a prokaryotic cell. But it isn't just DNA all by itself. It's complex with proteins. And the really interesting thing about archaea is those proteins are the same proteins that you see in eukaryotes like ourselves. They're called histones. But how many are packed in there and how they're arranged are different. And that's what this picture shows. So in our cells, right, we have several histone proteins, these little globule guys with the little hooks. And this one down here is also another protein. They complex with two wraps of the DNA. That's even coiled before that, where it's just one wrap, and you can see you don't have that histone 1 protein there like you do in eukaryotes. But, very similar characteristics. And then the vesicles that we talked about, like storing gas in those gas vesicles and the other ones that store sulfur and things like that. We find those inclusion bodies also in Archaea, where they will store stuff. And the really interesting thing about that is, okay, so you're storing stuff, right? Why is that beneficial to the cell, to store stuff? Probably the best example I can give is those plastic ice cubes. Do you guys ever see those plastic ice cubes? So you stick them in the freezer and then you put them in your drink and they don't melt per se into your drink like standard ice cubes, right? So like for me, if the orange juice isn't already cold, I don't really like to put ice in it. Why? Because it dilutes it, right? As the ice melts, it gets watery, right? And I like it not watery, right? So if it isn't always cold and they're wanting to put ice in it, I'm usually like, no, I'll pass. Give me something else, right? So is water a problem for these cells, you think? Remember, these are unicellular organisms. They're one cell. What happens to our cells if we put them, say, in a hypertonic solution where there's too much water outside the cell? What's it going to do? It's going to rush in and bust that cell, right? What do bacteria and archaea have that helps protect them from bursting like that? They have their cell wall. But that whole problem of stuff wanting to come rushing in is a constant problem for cells in truth. Especially if you're a unicellular organism living in an environment where it's not always going to be isotonic, right? We do the best we can to make an isotonic environment for our cells within our body, so we're not bursting them all the time. For them, this struggle is continuous all the time, right? Most of the time they're in a hypertonic type environment. They've got more stuff inside them than is outside, right? They're concentrating stuff on the inside. So that affects the movement of water, what we refer to as osmosis, right? In response to a concentration gradient. Water's always going to want to get into these cells, right? So how do we help with that problem? How can we lessen the amount of pressure of water wanting to come in? We got to have less stuff inside, right? Well, you need that stuff that's inside, right? Or you might need it, right? So that's why you're storing it, right? Because you're like, I might need this later. I don't want to just let it go. Because then, that's how some of us become pack rats. For fear that we might need it later. For microorganisms, there's going to be a time they're going to need it. So they definitely don't want to get rid of it if they're going to need it, right? They want to store their sugars and stuff like that for in case they need it. But if they encase it inside a vesicle, right, in an inclusion body, or they encase it, say, in protein or even in lipid, it now is no longer in solution. So it doesn't affect osmosis. So it's like a plastic ice cube, right? It doesn't water down your drink because it stays inside the plastic. Inclusion bodies, their benefit is, of course, storage, but storage that does not affect the osmotic pressure on that cell. So it makes sense why they would want to do that, why they would want to package these things up into these inclusion bodies. So again, gas vesicles, they have more to them too in that they can give them buoyancy in aquatic environments. So I made a change to this objective, and I thought one of the changes was already there, but I don't know. I've got like too many versions of this document. I need to get better at it. But you need to be able to identify the composition and function. I'm not sure if I don't think the sheet that you guys had function. But most of the things we talk about, we want to know how they function, right? But we're not going to talk about the limited internal membrane structures found in prokaryotes. So we're just going to stick to the composition and function of plasma membranes in general. Now there's a whole section in your book, the title of it is cell envelope. But what is a cell envelope? What structures make up the cell envelope? So what parts are there to a cell? There's a cell wall. Do you think that's part of the envelope? Yeah. What else does a cell have? We were just talking about, what's all that stuff on the inside called? Cytoplasm. Is that part of the envelope? No. What surrounds the envelope? What surrounds the cytoplasm? The plasma membrane. All cells have that, right? That defines the cell. Is that part of the envelope? Yes, that is the first layer of the envelope. So basically it's anything plasma membrane outward. So for most cells that is the plasma membrane. It could also be the cell wall. And as we've talked about previously when we mentioned gram stain and we're going to review today, gram-negative cells also have an outer membrane. That's part of their cell envelope. So that's a term probably most of you have not used before, huh? Hence why I literally defined it on our objective page. So most of us are familiar with what makes up a membrane, right? This first layer of the envelope. That plasma membrane made up of phospholipids in a bilayer, right? We commonly represent them by these little ball with two tails. So you had the hydrophilic phosphate group that loves water, right? So it's on the outside, and we know the outside environment is very watery, right? And so is the inside environment of the cytoplasm, very much a lot of water. So they get along, it all works out good. Problem with these fatty acids though, right? They're hydrophobic, they fear water. And so these actually will naturally arrange when you stick them in water, phospholipids, into my cells, where all the phosphates heads will be sticking out and all the fatty acids will be on the inside. Membranes get organized into two layers though, a bilayer. And then embedded within that bilayer are numerous proteins that have numerous functions. And that's really the functional part of a membrane. The phospholipids are pretty much just the barrier, right? They're the fence, the wall. The gates that we go in and out of, those are the proteins, right? What allows stuff in and out of that cell and really controls what happens for that cell. So the membrane defines that environment of a cell from the environment. So again, the workhorses are those proteins. So what are they doing? They control access of materials in and out, right? That's why we call it a semi-permeable membrane. Is only going to let certain things in and out depending on different conditions. And we are not going to go over in this class active diffusion, facilitated diffusion, antiport, symport. You should already know that. I'm not going to test you on it either though. So if you need to review, right, do so. But you know, you should have a general idea and you should remember from biology classes how specifically they move through those protein ports. So in addition, they capture and or store energy through photosystems, right? These exist in that membrane. So when you learned about photosynthesis, it happened in what organelle inside of a eukaryote? Photosynthesis. What organelle? No. what organelle inside of a eukaryote plant cell does photosynthesis? Photosynthesis. Can someone close the door? Where does photosynthesis happen in a plant cell? The chloroplasts. Oh my God, biology was like 80 million years ago, huh? Okay, chloroplast. What's stacked within that chloroplast? Thylakoid membranes. Whole stacks of membranes. So some photosynthetic cells, like cyanobacteria, they have more than just the plasma membrane. They got membranes on the inside of their cells. That's what my objective originally was pointing to. Within those membranes are those proteins that move stuff from one side to the other and make the generation of energy using light energy possible. Oxidative electron transport chain, right? You guys remember the ETC? Right? Where the electrons go. Where is it? It's in a membrane. In eukaryotes, what membrane stacked in what organelle? Do we make ATP? Yeah. Mitochondria. Remember those guys? In the mitochondria. Again, it's membranes with proteins embedded in it. And we're moving stuff from one side to the other, and we're controlling. Right? Those proteins are controlling who goes where and when, and that force of them wanting to go back to the other side generates energy. In this case, it's from breaking down other molecules. In the case of photosynthesis, it's light energy exciting electrons and moving them along. You can't have a chemical or electrical gradient without a membrane. Right? Just like in our cells, right? Those action potentials and muscular contraction, it's all possible because of membranes and ions moving from one side to the other. And then for them to be able to sense their environment. Some of those proteins aren't just moving stuff from one side to the other. They're actually sensing what's out there and sending information inward or releasing stuff outward. Because unlike us that have eyes and nose and ears so we can hear and smell and see and even taste our environment, for them it's all chemical interactions. They're always sensing via chemicals what's out there. Is it good? Is it bad? Do I want it? Do I need to run away? How is this possible? The proteins embedded in the membrane. So this is in more words than I just did. That other slide is more of a summary. But this is a term that we're going to use coming up, is proton motive force, commonly abbreviated PMF. And so that's using protons across a membrane to create the force, the energy needed to do something. Bacteria use this all the time to move their flagella. They don't generate ATP like we do to move flagella. They use proton motive force, which proton motive force goes one step further for us and our cells, and that we use it to generate ATP. So as I said, they detect, but they also respond to the environment. So energy happens on the membrane, sensing their world happens, right, via those proteins in that membrane. This is where it gets kind of interesting. Archaea have different membranes. Why would they have different membranes? They're prokaryotes, right? Bacteria are prokaryotes. They should all be the same, right? No, they have their own domain, don't they? So they're different in some ways, right? Definitely their ribosomal RNA, like Carl Woese looked at. But then when we look at other structures like their membranes, they're vastly different from both prokaryotes and eukaryotes. Why are they so different? Why do they need to be different? Depends on where they live, right? Remember, these are our extremophiles most of the time. Ones living in extremely hot or extremely cold or extremely salty environments. Does their membrane have to be able to survive in that environment? Absolutely. If it didn't, then they wouldn't exist. So a lot of these changes, these adaptations, you could say, have to do with this ability to live there, right? Some pre-organism formed a monolayer, which is more stable at warm temperatures. Doesn't fall apart, right? Because it's one layer instead of two. And that gives them that stability at that high temperature. This interesting guy seems to have an outer membrane and a periplasm space similar to the arrangement of gram-negative, and again, usually we use that term for bacteria, right? Their ATP synthesis enzymes are housed in that outer membrane, not the plasma membrane like bacteria. So why are they sticking it on the outside? I don't think we honestly have a hypothesis for that. But it's there, and there's obviously a reason on why it's there. It works for them in that environment. And so these differences, you know, if you read the toolbox in chapter 4, they're using this to help develop drug delivery systems and vaccination systems, these different membranes and how they respond differently in the environment of our body. They actually will stay together instead of fall apart. And then when we look closer, so okay, some of them form this monolayer, which, you know, makes sense. They won't come apart easily when it's really hot. But then when you look at this phosphate group, and the one down here, look, they're flipped. Boop. They're in the opposite direction. And then when you look chemically, the bond that forms between the phosphate group with the glycerol is an ester bond. But for archaeal membranes, it's an ether bond. And this is a similarity between bacteria and archaea. We both have ester linkages, right? That's how we chemically connect those two pieces, where it's a different bonding scheme for archaea. Again, probably to be more stable in those extreme environments. I don't expect you to be able to detect an ester bond from an ether bond. I still am trying to figure it out looking at these diagrams because it's been too long since I've had organic chemistry. What's the important take-home message here? One's an ester bond, ester, I don't know why I always say Easter, ester bond, and one's an ether bond. So, what are they? Two of these things are alike and two of them are not. I can't remember that song. They're different, right? They're different. That's what's important here, is they're different. One is an ether bond and one's an ester bond. But you see what I mean? Like, don't get caught up in the name of the bonds, right? Focus on the fact that they're different, right? Archaea are different from both bacteria and eukarya on how their phospholipids are structured. And even they can form monolayers, which both bacteria and eukaryotes do not do. We have lipid bilayers. You'll notice too, if you were to look at the, and I know this picture is kind of small for this, I probably should have blown this up. Maybe I'll just do that. If I'm having trouble, I know you guys are. So I just want to key in on maybe even, I want to crop this side too. On this one guy. Should I have left the other one for comparison? Maybe so. So back one more. I'll just cut the bottom one out. Because these two are the same except for the mono versus bi. Alright. Now let's look closely now at the fatty acid chains. Do you see this? Carbon can form up to 4 bonds with other molecules, right? It can share 4 electrons. So notice here, it's what we call saturated, this top one. Do you see how it's doing all the maximum bonding with hydrogen? Right? So saturated fats are things that we know are what at room temperature? Solid, right? Butter, lard. They're saturated fats. They say you should avoid those, right? Unsaturated fats are what at room temperature? Liquid, oil, olive oil, vegetable oil, right? Those are unsaturated. When something's been hydrogenated to make it solid at room temperature, like peanut butter, which usually has an oily component to it, right? But you usually don't see that because they add hydrogen and they make it saturated. The reason why unsaturated, which is what you see here, right here it's got some double bonding with the carbons, that gives it flexibility and mobility. All right, so it's able to move. So if you're in a really cold environment, you're probably going to want to be a lot of unsaturated, right? So you can move. Where if you're in a really hot environment, you want to be saturated so you don't completely fall apart. Because temperature is going to affect fats, as we know, because you can make butter melt, right? Just heat it up. Because you're changing the bonding structure. Now, when you look down here at theirs, you're like, whoa. What is that? What is that? There's more carbon and hydrogen sticking off the sides. That's called branching. So these guys are like really thick, right? Like this is like more than just saturated. I mean, that's saturated. This is a lot. Like really big molecules. So do you think this would help you out if you live in a really hot environment to have really thick fatty acids? I mean, this is like super saturated. What you think? Yeah. More protection, right? More hydrogen, more carbon, more molecular mass, less likely to melt and fall apart. It stick to itself. So Archaea have this different bonding, right, for hooking their fatty acids to the phosphate group. They can form monolayers, and they can even have branched fatty acids, which makes them like super saturated. All of these, of course, are adaptations to the extreme environments that they live in, like extremely hot and cold and salty, crazy. So is that it? That's it? Phospholipids, proteins, good, we're gone, we're done, walk away. No, one more component that can be quite important for some cells. Hapanoids is the name given to these molecules that are said to be sterol-like molecules. The sterol molecule that's embedded within our phospholipids we know as cholesterol. This helps give stability and rigidity to the membrane. You make cholesterol for your membranes, right? Bacteria make something similar. They make hapanoids. So when you look at the structure, here's cholesterol. There's the bacterial hapanoid, whatever. Oh my gosh, these names will kill me, right? But I mean, look at it. It's very similar, right? You have these planar, big ring structures, right? And some other components. A lot of similarities there. And a chemist would tell you the same. So they function very similarly, right? They help add rigidity and stability to the membrane. So we have cholesterol. You make cholesterol to put in your membranes. When you eat animals who do the same thing we do, right, put cholesterol in their membranes, then you get sometimes more cholesterol than you need, right? So then you have to do what? If you're eating too much, you need to eat less, right? You get high cholesterol. Or you could fall into the other category where your body genetically, unfortunately, makes too much cholesterol. So even diet alone isn't going to help you. But the good news is we've got drugs that'll help you out with that. So you don't get too much of this in the system and clog up your arteries. But unfortunately, we don't want to shut it off completely, right? Because then you couldn't make functional cells, stable cells. Right? So usually, like, when I say cholesterol, everyone's like, oh, that's the bad stuff, right? No, it's actually important, right? Just gotten a bad rap because we, sometimes don't do what we're supposed to. So the cell wall for bacteria and archaea, guess what? Are they the same? No, right? Obviously, we're going to have some differences for those special archaea, right? And of course, the most we know is about bacteria. So we're going to focus on that. We've already kind of looked at that, right? For the gram stain, we're going to kind of dive in a little bit deeper into the structure of the cell wall for bacteria. And then we're going to do a comparison between bacteria and archaea. In other words, look at the difference. There's an awesome animation in chapter 2, right, titled Peptidoglycan, that I highly recommend you take a look at. Peptidoglycan. So that's actually the chemical name for the cell wall structure in bacteria, is peptidoglycan. And as we've said, I said earlier, it's really important for maintaining the shape of the cell and keeping that cell from bursting under osmotic pressure. So it's made up of subunits, so different pieces put together, and actually the name itself tells you a lot about what it's chemically made out of, peptidoglycan. So peptido is the name of the bond between amino acids. So are there some amino acids in here? Yep. Glycan refers to sugar. So are there some sugars in here? Yep. So one subunit, right, and that many subunits get put together to form the wall itself, just like stacking cinder blocks, right? For peptidoglycan, the two sugars, which are modified sugars, are N-acetylmuric acid, which has a peptide chain attached to it, which means amino acids. About 4. And the exact amino acids will vary depending on the organism. Right? But in general, these are the most common ones. So some lysine, some alanine, and some isoglutamate. The other sugar is N-acetylglucosamine. So the sugars are alternating, right? So you have NAG, NAM, and then you start over, NAG, NAM, and it keeps going. And then hanging off of each one of the NAMs is the tetrapeptide. So then we start forming these linkages between even the layers. That's what the peptides, the amino acids, do. And even this can vary, how they join together. So for a gram-negative bacteria like E. coli, they bridge between these two in their layers, amino acids, where for gram-positives, because remember in comparison, who's got the thick layer of peptidoglycan, gram-positives or gram-negatives? Gram-positives. So Staph aureus is a gram-positive bacteria. It has an additional linkage of 5 glycines, what we call an intrabridge, between the two tetrapeptides. This creates greater stability of the multiple layers that form in gram-positive. Because they'll have up to like 30 layers, right, of this sugar. So imagine a jawbreaker, right, with all the layers. So we're talking about like 30 layers of sugar hooked together by amino acids. So the other interesting thing, and we'll come back to this later, you learn about in chemistry and organic chemistry, when we form these molecules, they can have mirror images of each other. Those are referred to as isomers. So it's the same atoms, but the way they're arranged around the carbon atom is mirror images of each other. So it's actually really interesting because the D form of amino acids is not common in nature. Most amino acids are in the L form, the left-handed form, instead of the right-handed form, which we denote D. It's interesting. It's like, how do they put this stuff together? Why are they making it different? You know? It's kind of cool. So then when we get to Archaea, notice one of the differences is this tetrapeptide. In bacteria, as I said, most of them are D. The amino acids are the D form, but for Archaea, it's the L form of the amino acid, the one that we see the most in organisms. And then notice still alternating sugars usually, but their cell wall molecule, I guess you could say, instead of saying peptidoglycan, is often referred to as pseudomurein. And so you'll see they have the N-acetylglucosamine just like bacteria do, but they don't have the N-acetylmuric acid. They have a different modified sugar, N-acetylallosamuric acid, abbreviated NAT. So this molecule is different, and the amino acids, right, are mirror images, isomers of what you see in bacteria. But achieves the same goal, right? It's a structurally sound cell wall. Helps with osmotic pressure. Helps hold their shapes. But chemically, right down to what molecules are making up it, they're different. Not huge differences, right? But they are different. Some archaea lack a cell wall. So what has to really help them out? Their cytoskeleton, right? Do we have some bacteria that lack cell walls? Yeah, we have a whole genus of bacteria. Do you guys remember the name of that genus? One of the members causes walking pneumonia. They can stretch out and kind of look like fungi. Myco for fungi, plasma for plasma membrane, right? Mycoplasmas. How am I on time? There's a few minutes. Okay. So next time we're going to look at how can we destroy a cell wall and therefore destroy a bacteria, right? Because if they don't have a functional good cell wall and they're used to that to help protect them against osmotic pressure, what's going to happen to them if we weaken their cell wall? They're going to burst and die, right? The osmotic pressure is going to kill them. So the cell wall is actually made inside the cell, right? And it's transported outside the cell. Right? So the pieces are made inside and then they're linked together outside the cell. This is important because when we talk about antibiotics next time, to interfere with the making of it, they actually go inside the cell and interfere with the machinery. So the first step for the antibiotic to be effective is it's got to get in, right? And things like E. coli are naturally resistant to drugs like penicillin because guess what? It can't get in. We produce substances that will degrade the peptidoglycan. Will destroy it chemically. So we produce lysozyme in our tears, our saliva, just about any secretion you can think of, our mucus. This enzyme breaks the bond between the sugars. And so basically chews up the peptidoglycan, so much so that the osmotic pressure gets to the cell and it bursts. This is a new one. This one's interesting. Lysostatin. This is an enzyme produced by, I think, another bacteria. I'm not sure. But it attacks that intrabridge that's found in gram-positive bacteria like Staphylococcus aureus. And that's why they named it lysostatin, staphylin referring to that it attacks Staphylococcus bacteria, these bacteria that have this intrabridge. So it's breaking apart the pieces of the intrabridge between the tetrapeptides. Oh my God, that's a mouthful. So next time, as I said, we're going to come back and we're going to talk about these antibiotics and how they work.